all of you know Kate, and at the beginning of this, I didn't know Kate. She was coming from Hatfield in the Fisheries and Wildlife Department, where she was just finishing up a master's looking at flatfish out in coastal oceans. So very different from the type of ecosystem that I was familiar with. And prior to that, she was in Northwestern, urban area up in Chicago. And I think it's safe to say that we completely converted Kate into being a desert rat. Is that safe?
Nice work, yes.
I think it was a willing conversion too. And as you'll see from her talk, which takes place in desert ecosystems in the southwestern U.S., at the time that Kate came on board, my lab group had been primarily looking at the evolution of behaviors and life histories in just a handful of organisms. And a few of my graduate students were just starting to open that up and looking at the whole community surrounding those evolutionary processes and traits, and starting to look at a lot of the other aquatic insects that are present there and asking questions about what that all means. And Kate came along and essentially took that to a new level, where not only did she look at the community level context in these desert streams, but she has really moved forward in looking at what each species means. So she's converted a lot of the species into what we call traits that allows you to ask questions about the functioning of ecosystems in the context of these environments that have a lot of flash floods and droughts and other extreme events like that. And so Kate's really pushed forward those methods and some of the theory based on that as well. So she's really kind of broken into new frontiers as far as our lab goes. And again, those of you who know Kate know that she's extremely well organized and focused. And in fact, I think one of our first meetings, she comes in with an agenda, sits down, and says, okay, here's what we're going to talk about in our meeting. I was like, what do you expect out of me for a PhD? And, those of you who know me, I'm not, it's minute for minute, okay, a lot of time. So I just sort of think about this, and just sort of off the cuff, I'm like, well, okay, maybe a paper already accepted, a couple in the review process, and I'll give you like one kind of wild card paper that's, maybe really interesting and, maybe a little bit harder to get into journals. And sure enough, I thought about it this morning, and that's exactly where we are. She went right to where she said she would be, and I think is delivering that really well. And in addition to that, I think those organizational skills have allowed her to be extremely successful at funding her own research. She has written for and received many fellowships, many small grants, travel awards, you name it. She's pursued it and nailed it, so she's an extremely good role model for being successful in that. And beyond that, she does excellent science. And it's a lot of fun to have in the lab. And so I'm going to stop now and let her show you some of these wonderful things she's been working on the last few years. There you go, Kate.
Great. Well, thank you all for coming. This is a long time coming today, and I'm really excited that you all are here to share it with me. I'm excited to tell you what I've been up to. So today, the title is Aquatic Community Responses to Drought Disturbance, Experimental Manipulations of Top Predator Extinctions and Stream Time. And this charismatic critter here, the giant water bug, will be a focus of a part of my talk. But from there, we'll expand and talk about ecosystems and how to apply my science in your own system. So please, throughout this talk, I'll show you photos of my system and how amazing I think it is and how it's beautiful and awesome. But I know you all have those systems in your head too, so try and keep this in that context. So we've heard a lot about extreme climatic events in the news lately. This top photo is a forest fire in California just this summer. And this photo here is the Colorado floods from last month, devastating damage, lots of property, few human lives lost. And on the right-hand side is a stream that has gone dry. This is a stream in northern Mexico, and 15 years ago it flowed. And now it's dry and just a cobbly stream bed. And so the impacts of these disturbances on human communities are readily apparent and in the news all the time. However, scientists are beginning to ask this question: how do these changing disturbance regimes affect ecological communities. And in fact, the Intergovernmental Panel on Climate Change, the IPCC, their report they came out with last month again confirmed what we've all suspected, that the magnitude and frequency of these extreme climatic disturbances are increasing and are predicted to continue to increase into the next century. And so when we think about how these changing disturbance regimes affect ecological communities, the first thing that comes to mind are these direct pathways. If the climate gets hotter, the climate gets drier, forest fires, those all do massive damage to communities of organisms. But there are both direct and indirect effects of these disturbances. Here's the pathway we just talked about. Climate disturbances directly affect ecological communities, but it's also possible that some other factor can be mediating those effects. So this is an indirect pathway by which climate disturbance can affect those organisms and communities. And so today I'm looking at one indirect effect specifically. And it's one that I believe is going to have a huge effect on communities. And that is top predators as mediators of climate change. And so most of you in the room are familiar with this traditional food chain. Here at the top, I have a wolf, a top predator, then a coyote, mesopredator, a rabbit herbivore, and some grass as primary producer. And these are frequently called trophic cascades. And you can see because the effect of changes in the abundance or distribution of any of these animals is going to affect all the levels below it. And so when that top predator, when it goes locally extinct or when its populations are impacted, we frequently see these dramatic increases in the abundance of mesopredators, decreases in herbivores, and increases in primary productivity. So in addition to trophic cascades, these are sometimes called top-down effects. And you can see why. So when we combine this really important role that top predators have in communities with the fact that top predators are disproportionately vulnerable to these disturbances, and this has been documented in many ecosystems with many different top predators, then this becomes this sort of perfect mix of bad things that are happening, an imperfect mix, maybe. So my question today is, does disturbance indirectly affect communities through top predator extinctions? And so to address this question, it really helps for me to have a model system that can represent this extreme abiotic environment, top predator extinctions, and complex ecological communities. And so for those of you who know what I do, you know that I've chosen fragmented arid land streams as my model system. So I work in the deserts of southeastern Arizona, and this region is called the Madrean Sky Islands. So on this map, each of these green blobs is a mountain range. And those mountain ranges are surrounded by a sea of desert, hence the name Sky Islands. So all of the remaining aquatic habitat is in these headwater streams that are isolated in these fragmented mountain ranges. Back in the Pleistocene, these streams used to flow into large desert rivers through the river valleys. But since then, things have become much drier, and now we're limited to these mountaintops. So in addition to that fragmentation, these streams are fragmented from each other because they no longer connect here. So we have fragmentation at the landscape level and within mountain ranges. These nested scales of fragmentation become even more complex when we look within a stream, because we not only have fragmentation at those other levels, but we also have fragmentation of individual streams. This is a dry season pool, and this serves as an aquatic refuge for animals through the dry season. There's a very extreme dry season in these regions. And so these remaining aquatic refuges are extremely important. Now, given increasing prevalence of drought disturbances, we're seeing more of these pools dry, like this one. And the loss of that aquatic refuge in an entire drainage causes local extinctions of organisms. So we definitely have this severe abiotic disturbance, and then drying in this region results in the local extinction of this aquatic invertebrate top predator. This is the giant water bug, and I'll be discussing it more later. But this is a flightless insect, and therefore it's highly threatened by these drying refuge pools. So the system has both of these components. But it's quite easy to look at that picture of two of my co-authors over that little mucky pool and think that those can't contain complex communities. And so this isn't an effective model system. Here I am peering into a stagnant pool, and when I look below the surface, there are up to 150 different species of aquatic invertebrates. This is an incredibly diverse system, and they have amazing adaptations to let them survive through this harsh, dry season. So we've got all three of these components to be able to mechanistically address this question. And I'm going to do so today through manipulative experiments. I'll be addressing the direct pathway and this indirect pathway that's mediated by top predator extinctions. So my talk today is split up into 5 parts. In part 1, I'll examine that direct pathway through a drawing manipulation experiment. And in that experiment, I set up artificial communities, and then I manipulated drawing and saw how those communities responded to disturbance. In the second part, I'll describe another manipulation experiment to examine that indirect pathway. And that will be an artificial top predator removal study. Then I'll take that information and use it to create a disturbance traits framework that may be applicable in other ecosystems. I'll apply that framework to my model system using data from part 1, part 2, and a field data set that my lab has already been publishing, and then try and pull it all together at the end. Now, I'd like to tell you now, I will not be spending much time on the first 2 parts. These are both in revision and should be in press by the end of the year, if all goes well. So you'll be able to read about them. And I've also given talks on them probably 8 or 10 times on campus already. So if I put these different parts of the talk on this diagram, we're going to start with part 1, with that direct pathway. So this picture here is of a mesocosm. And mesocosm is just a technical jargon term for an artificial community that's supposed to replicate those pools in the field. And the nice thing about bathtub-sized pools in the field is that they're very appropriately replicated by my mesocosm tanks. So I created a bunch of mesocosms, and I inoculated them with invertebrates from arid land streams in southeastern Arizona. So I went out and I sampled a bunch of streams, different habitats within each stream, and I allocated those communities evenly across the tanks. Then I applied 3 treatments. A control in which I maintained the water level at 10 centimeters, moderate drying in which I permitted the tanks to dry down to 7 centimeters, and then a severe drying treatment in which those tanks desiccated to 1 centimeter of water. These mesocosms were covered to prevent dispersal and colonization, so the only invertebrates that were in there were the ones that I put there in the beginning. The experiment lasted 6 weeks, which in this region is about the duration of that summer dry season. And then at the end, I sampled the communities and identified the invertebrates. And so on this next slide, I'm going to show you some of the results from this first experiment. Here on this figure, this shows species richness, or the number of species that were present in each of those treatments. And then here on the x-axis, I have control, moderate, and severe drying. So there were no differences in the number of species in any of those treatments. And this was really surprising. This severe drying treatment contained only 1 centimeter of water. Now that means wet sediments and little dimply pools of water. So it's really incredible that the same number of different kinds of species were present. What was even more incredible, that there were no significant differences in community composition. So not only were there the same number of species, but they were exactly the same species in each of these treatments. The only thing that changed across these treatments is the abundance of invertebrates, which decreased between the control and moderates and the severe. And along with that decrease, the density of invertebrates increased. So from this first experiment, I found very high resistance to stream drying, higher than I'd suspected. And then these no changes in species composition. I need to point out, though, that I did not apply a complete drying treatment in this study. So this may not be an appropriate representation of what stream drying looks like in the field. I was studying that drying process that precedes complete drying. So from here, let's move on to my second manipulative experiment, where I simulated top predator extinctions. So here's a picture of this invertebrate top predator. This is a hemipteran, and so it's a belostematic bug with the scientific name, Abetus herberti. The common name is the giant water bug. These insects are about 3 centimeters long, which is giants in these communities. And the important things for you to know about this species are, one, that it is flightless. You can see some vestigial wings here, but this species has completely lost the capacity for flight through evolutionary time. It's also important to know that it's an aquatic obligate. So that means it requires water for all phases of its life cycle. So you might not be surprised that it is highly vulnerable to local extinctions. And in fact, my lab has recorded 3 local extinction events from entire stream drainages just in the past 10 years in southern Arizona. The other really important part to know here is that it's the top predator. Here is the piercing mouth part of the giant water bug and the raptorial forelimbs. And so this sit and wait predator can sit and wait until some susceptible prey swims by, latch on with those raptorial forelimbs, insert the beak, insert digestive juices, wait for those digestive enzymes to do their work, and then slurp up the digestive remains. So not only is this really awesome, but it's also ecologically important, because it means that this insect can function as the top predator. And it can consume prey items that are much larger than itself, including both invertebrates and vertebrates. Okay, so we know that, and so we're going to use this threatened top predator for my top predator manipulation experiment. So here are a number of mesocosms. And again, I inoculated those mesocosms with invertebrates from the region. I applied 2 treatments, a control with intact communities, and then a top predator removal, where I simply removed the top predator from those tanks. And the predator is fairly conspicuous, it's large, and so it's easy to manipulate and remove by hand. Mesocosms were open to dispersal and colonization, unlike the previous study. This experiment also lasted 6 weeks. At the end of the experiment, I sampled the communities, identified all those invertebrates, and this time I repeated the experiment in 2 years. And those were 2 years with very different environmental conditions, because I wanted to see if the patterns I saw in 1 year held true for the following year. So in this case, I did see effects of this biotic disturbance on communities. Although, surprisingly, I found again no effect of treatment on species richness and no effect of treatment on species diversity. Where I did see an effect was this significant top-down effect on community structure. And I'm going to talk about 2 different aspects of community structure that are particularly important for our story. The first is with the mesopredators. So mesopredator, in my system, is just all secondary predators that are larger than 10 millimeters long. And this includes both larval insects that can't fly, and also adult insects that were able to move from mesocosm to mesocosm in my study. And because they could move around and my tanks weren't covered, they were able to colonize during the experiment. So here's a figure that shows how mesopredator diversity, the number of different kinds of mesopredators, changed when I removed top predators. So here's a control treatment and a removal, and this is for 2010, over here 2011. And so here you'll notice when I removed top predators, the diversity of mesopredators increased. And in 2011, there was no significant difference, but there was still a suggestion of a possible pattern. So I also found strong effects on the detritivores. And in this case, the effect was the opposite direction. So when I removed top predators, the abundances of this important numerically dominant detritivore were reduced. This is Phaloicus mexicanus, the sycamore caddisfly. And it's called the sycamore caddisfly because it builds its case out of sycamore leaves here. And here it is. Here's the insect nestled in sort of a sleeping bag of leaves. And this is really important. This is one of the few insects in these summer communities that will convert coarse particulate organic matter into fine particulate organic matter for the rest of the detritivores to use. So this was an alarming reduction that accompanied my simulated top predator extinctions. So when I combined the results of parts 1 and 2, these experimental manipulations, I found that there was really high resistance to drying disturbance, even extreme drying disturbance. But in the top predator removal experiment, I found low resistance to top predator removal. So both of these studies measured communities. I identified those insects and looked at how that community of insects changed. But what do these different terms mean? First of all, resistance in an ecological context usually means the maintenance of taxonomic composition. And so this is what I was measuring in that the drying manipulation experiment, because those insects couldn't leave and couldn't colonize. But the top predator removal experiment was also quantifying resilience, which is the recovery of taxonomic composition following a disturbance. But what do these resistance and resilience tell us about the resistance and resilience of ecosystems? Not the identity of the species, but the actual functioning in the environment. And to talk about that, species identity becomes less important than the function that each of those species is playing in the environment. And so I'm going to move on to this idea of functioning in my third part. So for this third part, I'll be talking about functional traits. And functional trait, again, is just jargon that means the characteristics of the biology of an organism that are associated with its functional role. So these could be things like gape size, how big the mouth is, because that'll affect the functional role of what kinds of prey it can eat. In my system, this might mean dispersal capacity, from this flightless insect to an insect with weak flight, to an insect that as an adult can travel kilometers from habitat to habitat. This could also involve even simpler things to measure, like the length of an animal's body or its biomass. So why am I interested in these kinds of things? One of the main appeals for me is because traits transcend taxonomy. So here's a stream in Australia. Looks really similar to the kinds of streams that I study in Cave Creek in Arizona. So if I go out with my aquarium net, and I walk up to both of these streams, and I dip that aquarium net in, and I get a community sample. So I have this really awesome entire food web in both of these samples. And those food webs look really similar. They both have invertebrate top predators. They both have a bunch of beetle and bug mesopredators. But they don't share a single species in common. They share a lot of other stuff in common that's probably important. So we need a way to talk about those systems and compare them that doesn't have to do with species identity. So I propose that I can use all this great taxonomic knowledge that Dave Lytle's lab group has been developing for the past 10 years in this system, and I can use it to create hypotheses about functional responses to disturbance. So how these functions of the community change, not just how the members of the community change. And then ideally, I'd like to see if there are inferences that are relevant to conserving these threatened habitats. So to do that, we need some new tools. In order to understand functional composition, we can sample a bunch of invertebrates and measure functional traits for each species in those communities. So for example, let's say I measured metabolic rate and drought resistance. So then I can make a figure here that has, say, metabolic rate on this axis and drought resistance on this axis. And then I can plot each of the species in my community as a single point on this axis. So say this mayfly is at this position because it has this particular value of metabolic rate and this particular value of drought resistance. So then, points that are closer together are species that share similar combinations of traits. This is called functional trait space, and I'll be referring to it as trait space from now on. So with the beauty of advanced mathematics and ordination methods, now I can add a bunch more traits. I've got a dozen traits, and I can simplify those traits and minimize the variability to create two axes that now represent a whole bunch of traits combined. So I'm about to present my functional trait framework to use in the context of disturbance. And this builds on a lot of work that's been done, both historically in community dynamics, but also recently in this sort of new burgeoning world of functional ecology. So in this world, we can sample undisturbed and disturbed communities, and then measure the functional traits of species in both of these communities. So here's my trait space again. And now I quantify, these are all species in the disturbed and in the undisturbed. And so now you'll notice that none of those points overlap. So that means that none of the species were in both the disturbed and undisturbed communities. So the most likely explanation for this is that there was species turnover. That disturbance was so harsh that none of these trait combinations made it. So for the field ecologists in the room, you know that this is kind of unlikely. That really, when you go out to the field and there's a disturbance, some of those taxa from before the disturbance will still be there after. The really tough ones, right? And so it might be more realistic that we would see an ordination that looks like this. Here, we have several points that overlap. So those are species that were in both communities. Now in this particular example, the blue circles occupy more space on the diagram than the red circles. This means they had a wider trait diversity. And so this is representing sort of a classic ecological prediction of the environment as an environmental filter, because the only ones that could survive after the disturbance were these invertebrates that had particular traits, the traits that let them survive harsh environments. So now, my goal for this section is to propose ecologically informed hypotheses from all the knowledge about our system, then choose quantitative methods to distinguish between these hypotheses, challenge the hypotheses with data, interpret results in light of my arid land stream system, and then generate new hypotheses. So you may recognize this. I am in fact proposing this slide. The reason this is novel is because functional diversity methods are rarely applied in a rigorous hypothesis testing framework. This field is growing so rapidly that every other week some new metric to measure trait space comes out. And the current approach is to look at all that cool math and pick your favorites and throw them at your community data and see what sticks. And I'm suggesting that we return to this hypothesis testing framework. And that's another suggestion that's been echoed in the literature. Okay, so now I'm going to propose 6 hypotheses that are how I believe my communities may respond to disturbances. So here's the trait space again. And now I have 5 species, that's 5 points in trait space. And now I can code the size of those circles by the abundance of that species. So here I have 5 rare species and 5 abundant species. And then just like before, oh, I was supposed to mention this little circle inside another circle, that means those 2 species are functionally redundant. So they share exactly the same trait combinations. So here I've done that for disturbed and undisturbed communities. So my first hypothesis is the null hypothesis. This shows that the red circles and blue circles completely overlap. This means that this community is highly resistant to disturbance, highly functionally resistant. And this may occur in a system such as my own, that's frequently disturbed, where you may expect those organisms to have great adaptations to withstand periodic disturbances. In sharp contrast to that is my second hypothesis, the directional change hypothesis, where only these 2 species are shared in the disturbed and undisturbed communities. And so we can visualize this if we plot these 2 x's. These are the functional centroids. So this is an abundance-weighted mean trait value. So that just means that you look at all those traits of all these species, and you sort of come up with a composite average. And then you do the same thing with the disturbed community, and you look at the distance between those x's. But there are lots of ways that communities could be affected by disturbance that don't involve a shift in functional centroid. One of them is represented by my third hypothesis here. This is the convergence hypothesis, where in the undisturbed communities, they have high trait diversity. And in the disturbed communities, that trait diversity is lower. And this is similar to that ecological filter idea we saw before. The opposite is the divergence hypothesis, where the undisturbed communities occupy less trait space. Now this could occur in a community where disturbance eliminates a competitively dominant species and opens up niche spaces for other members in the community. It could also happen if an invasive species arrives without supplanting the native taxa. my fifth hypothesis, the equal impact hypothesis, was kind of similar to the null hypothesis, because all of the species exist in both communities. However, all of the species abundances are lower in the disturbed communities, because all the red circles are inside the blue circles. So this is an equal impact across all taxa, and this could happen in a situation of extreme habitat loss. Say you have a rainforest, and a part of the rainforest is clear-cut, but you still have an intact part left. So all those species may be affected equally to that disturbance. And my final hypothesis, the sixth one, is the trickiest to understand. So this is the skewed impact hypothesis. So first I want you to notice that there are blue and red circles at every point in the diagram. So all those species were present in both communities. But the size of the circles is distributed very differently. So this means the abundance distribution differs. And so these circles may be larger, meaning that these trait combinations were better suited in the disturbed communities, and vice versa for the undisturbed communities. Now, if this is extreme, we may still see differences in the location of that centroid, because remember, these x's are abundance weighted. But this hypothesis would detect signals of decreasing abundance that may precede local extinctions of taxa. So this hypothesis may be very important as we're thinking about conservation of these ecosystems. Okay, so now I have these 6 hypotheses, and these are non-mutually exclusive hypotheses. So communities that are disturbed may exhibit multiple hypotheses at once. So I told you the next step then is to take my hypotheses and choose these quantitative methods to be able to tell these apart with my own data, right? So there are now over 30 quantitative ways to calculate differences in trait space, to calculate functional diversity, and it seems like a new one is published every week. So given that there are so many, you may be surprised that I didn't find one that I liked. And so I had to make up my own. So here's the reason. I called it functional distance. And this metric is designed to answer this question: does disturbance affect the distance between those x's, between the functional centroids, right? So that sounds like an easy question. Here in my map, my ordination, you see the 2 x's, and it's fairly mathematically easy to measure the distance in multivariate, multidimensional space. However, all good scientists replicate their experiments or take multiple field samples. So this is just one undisturbed community and one disturbed community. So if I have a whole bunch, and these are unpaired community samples, like my mesocosm experiments, some I removed top predators, some I didn't, then that means I have to compare the location of these centroids with all the centroids of all those other disturbed and undisturbed communities. And so to do that, I use generalized linear mixed models. And so these use community as a random effect to account for the non-independence of those pairwise comparisons, those pairwise distances. And this is a technique that's frequently used in genetics to calculate pairwise distances, pairwise genetic distances. The second metric I'll be using is functional richness. And functional richness sounds complicated. The multidimensional volume of a convex hull around all species in trait space. All it means is that you take the volume of a big cube or multidimensional space, big basketball drawn around all those species, and you compare that volume with the volume of the other treatments. And you see if those volumes are different. Who occupies more trait space? And then I can simply test those between my 2 different treatments. The last metric I'll be applying is functional dispersion. And so this one, I specifically chose to address this question of how the abundances are distributed. And so here, I can use the abundance-weighted distances of each species from that species community centroid. So it's kind of like connector dots. We draw lines from all these different species to the blue X, and the same to the red X, and then compare them for each replicant. Okay, so now comes the fun part. I get to apply all of that theory to my own arid land stream system and see if any of this is worth my time. So the goal for this section is to determine how drought disturbances affect aquatic and vertebrate functional diversity in my arid land streams, right? Fairly simple question. And in doing so, I want to test this whole framework I just showed you. And ideally, I want to see if it provides any inferences that could help conserve. So I'll be applying applying this framework to the two studies that you've heard about now, the drying manipulation and top predator manipulation. But I also wanted to include a field data set. And so, luckily, my lab has been doing studies like this for a while. And I chose one particularly exemplary study from French Joe, teeny tiny stream called French Joe. And so, French Joe is located in the middle of this Madrean Sky Islands range. This X over here is where I conducted the manipulation experiment. And this X here is where my two co-authors conducted this field study. And so Mike Bogan and David Lytle went out to this stream, and they sampled it for several years in a row, 3 years in a row. And this is one of those streams where here I'm just showing you a single pool that remained during the dry season. This is one of those refuge pools that's really essential for those communities to last to the following wet season. So they sampled it for 3 years in a row, and they went back in 2005, and it was dry. And they have lots of evidence, sociological, from naturalist notes, from the biological community, from the plants, that this hadn't dried in a really, really long time. And so they continued sampling for 4 years following that drying event, because the water returns every wet season. They found extirpation of 6 species, including our giant water bug top predator. They found no changes in species diversity, though, surprisingly, which means that a lot of those species were replaced. They found significant shifts in community structure with no recovery after 4 years. And the important part for me is that this field study recorded concurrent drying and drought-induced top predator extinction. And really, this is the study that motivated my entire dissertation. Great paper, it's award-winning in freshwater biology, captures this real-time drying event. So in order to apply this fancy framework, I needed some traits. And it is totally unrealistic for me to measure everything on all 100 or 150 species in my system. So lucky for me, people have been assembling trait databases recently. And some of my lab members have even been assembling them in this very system with these very insects that I had. So I had a whole bunch of traits I could choose from. So I selected 7 traits that I picked because I thought that they would have something to do with drought resistance. The first 2 are associated with trophic status. This is body size and functional feeding group. So functional feeding group just means it's a combination of what an animal eats with how the animal eats it. So it's like the sort of summary trophic variable. The next 3 traits were associated with movement. Locomotion, dispersal, and respiration for aquatic invertebrates is a movement trait, because whether or not they can breathe air or have gills really determines their flexibility to move around in the landscape. And the final set is diapause and volatinism. These are life history characteristics. So diapause is when an insect can sort of create, it can insist. It's kind of like hibernation in mammals, where it can just hang out when the water goes away until the water returns the next time and it hatches out and becomes a happy aquatic invertebrate again. And then volatinism just means the number of different generations that an insect has in a year. So I took this and I applied it to my data. First, the drying manipulation experiment from part 1. So along the bottom here I have all these hypotheses that I told you about. And just to remind you, in the taxonomic analysis I found no differences in species richness across any of those treatments. And so I had predicted that I would see no change in the functional space. And I predicted I'd have support for the null hypothesis. So, this is what I found. Don't be scared, it's okay. So each of these circles is still a species, right? And the blue and the red are still undisturbed and disturbed, where drying is the disturbance. And so the important thing to notice here, first, is that my convex hulls, that area, they're pretty much the same for the blue and red lines. So not a lot of difference in trait space. But what I do want you to notice is that most of the time the red circle is inside the blue circle. So this means that the abundance of each of those species decreased following disturbance. And there was no difference in any of the other functional diversity metrics. So in fact, this decrease supported my equal impact hypothesis. Okay, the top predator manipulation experiment. So in this case, the disturbance was biotic. The disturbance was me removing top predators. And just to remind you, I found changes in community composition, but no changes in richness, and some species replacements. So I had predicted a shift in the functional centroid because of those species being replaced, but I didn't have any reason to think that there would be changes in any of these other hypotheses. So here's what it looks like. First, I want you to notice that those disturbed communities, the top predator removal communities, occupied more trait space than the undisturbed communities, which means they had significantly higher functional richness. So that supports my divergence hypothesis. I also saw a shift in functional centroid and an increase in functional dispersion. So you may be looking at this and saying, this whole movement of the axis thing, that doesn't look true here. Now remember I told you that each of those functional centroids is abundance weighted. So that means, here, if we look at how big the circles are, and we notice that the blue circles are pretty much the same size across the entire functional space. Maybe they get a little bigger over here. But we can compare that with the abundances in the disturbed communities, and we see that they're mostly located around the periphery of the distribution in this half. So those abundance differences were significant enough to pull the centroids apart in trait space. And that's why I found support for both the skewed impact hypothesis and directional change hypothesis. Okay, so let's move on to the field data set. Mike Bogan and David Lytle found changes in community composition, but no changes in species richness. And they again saw these species replacements. So again, I predicted a change in centroids, and I also predicted convergence. Because drying is such a harsh filter, I thought that it would limit the types of trait combinations that would be able to survive following stream drying. So here's their data. Now you'll notice that the blue circle is bigger than the red circle. So this means that there was a decrease in functional richness after stream drying, and support here for the convergence hypothesis. But there was no change in any of these other metrics. So I saw a lot of these made sense. A lot of these patterns were sort of consistent. However, there was one that was confusing. And that's this one I've shown here. Here's the top predator manipulation, ordination, and here's the French Joe field study again. So, if you recall, the whole reason I was simulating top predator extinctions was because in the field, we're seeing top predator extinctions. And in the field, this top predator extinction was associated with a reduction in trait space, functional convergence. But in my experiments, I saw the opposite. I saw functional divergence, which seemed like this huge contrast to me. But then I remembered how important the mesopredators were in my taxonomic analysis. So I figured out where they were on this ordination, which of those dots were the mesopredators. And I found that they were these taxa around the outside of the ordination space, these colonizers that showed up following top predator removal. I can contrast that with the colonizing mesopredators in the French Joe field study, which all had very homogeneous trait distributions. And this makes sense when we look at who the colonists were. Here in the top predator manipulation, it was this diverse combination of dragonflies and damselflies and dobson flies and bugs and beetles and all different traits, all colonizing those tanks. But in the field study, those colonizers were mostly beetles and bugs with a very narrow range of trait combinations. And this makes sense when we look at spatial contrast. So we know that the colonist source must be important, right? And if we recall that the field study was conducted in this teeny tiny mountain range, and both of the manipulative experiments were conducted in a large mountain range that's full of perennial streams. So here's a map of some of the perennial and intermittent water where the manipulative experiments took place. Here's the manipulative experiment, and it is 100 meters from the nearest perennial stream. When we contrast that with the spatial context of the Bogan and Lytle field study, it's 9 kilometers from the closest perennial water. So any invertebrates that recolonized had to do so 9 kilometers over a mountaintop divide. So that's going to really homogenize the types of traits that are going to get those bugs there. So when I pulled together parts 3 and 4, I hope I demonstrated that experiments are needed. We had this field study, and I could differentiate between those direct and indirect effects, and also between the biotic and abiotic effects. Because in nature, all these things are happening at once. And then I could use that information to select functional diversity metrics specifically to address my hypotheses about the system. And I created this rigorous hypothesis testing framework, and it yielded new insights. This importance of spatial context, the importance of colonist sources that we hadn't noticed when we were just looking at taxonomy alone. So when I bring it back to the system that I care about, and think about how this may apply to arid land streams. I found that these insects are highly resistant to extreme conditions. This is an ecosystem that's frequently disturbed. And disturbances that don't seem like disturbances here would decimate other aquatic systems. They've evolved amazing adaptations to let them survive. However, they have low resistance to disturbances such as complete stream drying and top predator extinctions. And so this is particularly concerning because we know that they're happening at once. These two things that are dramatically affecting communities. And this is even more concerning when we go back to that IPCC report and we think about these predictions for the future of an increasing magnitude and frequency of extreme events. And I experienced some of this increasing magnitude and frequency even during my PhD. This photo was taken in 2009, where I tried to do one of those manipulative experiments in the actual stream. And an unseasonably early and unseasonably severe flash flood destroyed the experiment. Here's a photo from my 2011 field study, in which a catastrophic forest fire destroyed 350 square miles of Chiricahua mountain range. That's almost the entire mountain range was burned. And this is after decades of fire suppression. So I do think there's some management implications of my work. This photo here is Vegas, which is not in southeastern Arizona, I know. But very similar things are happening in Vegas as are happening in Phoenix, as are happening in Tucson, as are happening in Texas, in all these arid land systems. So I couldn't get the 2013 version. But this is the population growth that has just happened between 1973 and 2000. And all of those people use water. And so we could use my information to time water abstractions better. So we are not draining down the aquifers. And new evidence has finally documented, yes, you drain down aquifers and those springs at the top of the mountains stop. They are directly connected. We also may be able to create refuge habitats that are scattered throughout the landscape to provide those recolonization sources. But more important for me than management implications, I'm hoping I'm communicating today my passion and amazement at these incredible systems. So you can hike for miles across the desert terrain, cacti, and you hike, and then you get to these incredible aquatic habitats that are in the middle of nowhere, that are totally hidden in the desert. And you find habitats like this, with these little gems of biodiversity. And so I like to think that they have some hope, and that if we take some active steps, we can prevent these habitats from disappearing altogether. So I'd like to thank my co-authors, first of all. I was told that I should use I for my dissertation defense talk, but I think the entire audience knows that this is a we. Especially I'd like to call out Laura D. and Steve Miller, who are my collaborators at UC Santa Barbara, who couldn't be here today. I'd also like to thank my committee members who've been incredibly supportive. Other supports, including Tracy, Tori, and Tara, and my dissertation would not have existed without them. I'm confident. This is our office staff in zoology, and I'm really grateful for their assistance. I need to say just a quick thing about the other half of my PhD. I showed you one half, and it was a really exciting, rewarding half, but this was the other half. My involvement with the Women in Science group on campus was extremely rewarding, and my teaching career at OSU was extremely rewarding. And so I need to thank all of these people, all of my students that continue to inspire my science. Yes, my science inspires my teaching, but my teaching also inspires my science. It goes both ways. I'd like to thank my mentors. So I encourage all of the graduate students in the room to get some quick. These can be your friends, these can be your colleagues, these are people who are going to believe in you when no one else does. You need them, and you should research them for you. I'd like to call attention to one in particular. Heidi Fisher is a science writer at Arizona State University, and she opened up her home to me for three-month-long field seasons in a row for free because she believed in my science. And she's in a hospital with West Nile virus in Phoenix today, and so she couldn't be here. So, thanks to Heidi. I'd like to thank the Lytle Lab, whose shenanigans have been so awesome and keep me grounded. Thank you, folks. I'd like to thank my friend group in Corvallis. And you're not all up here, because I didn't have enough space, but thank you. I'd like to thank my family. Paul and I calculated last night that I am in the second week of the 25th grade. And I am really, really grateful to you for getting me here. Thank you very much. And I'd like to thank he's been a great inspiration, and he believes in me, and that means a lot. And with that, I'd like to take some questions if I have time. Thank you.
Before we get going on questions, I want to let everyone know we're going to have an event, sort of a barbecue potluck over at my place at about 5 o'clock today. So I'm going to write you the rest up on the board.
Everybody's welcome. Jesse. So the question is about looking at individual traits. And I've looked at them all combined in this weird multidimensional way. But if I took single individual traits, could I see how those are affecting ecosystems? And that's a whole separate field of ecology. And I wrote a proposal to work with a guy in the UK to look into that, to combine the univariate direct effects with this multidimensional way that I approached it. So ideally, the future of functional ecology is to merge those, but they're completely separate literature right now. So, no, I haven't done it. Yeah.
First, that was a really great talk. It was really complete and articulate. So you started out talking about trophic cascades, and you gave this sort of hint that there may be something going on with the trophic cascade. As you remove that top predator, you change the abundance of a shredder. Have you guys gone in to look at sort of that next level down, either CPOM or FBOM abundance in these systems to see how that has changed as you lose your top predators?
No, I tried. I made an attempt at leaf litter bags to record decomposition rates, and I worked a little bit on getting a chlorophyll A, but I just didn't have the time or the resources to make that happen. It's definitely the next step that needs to happen in this research.
Just anecdotally, do you think it's happening?
Yeah, so I have this great photo looking in predator removal, and you look down and the predator removal tanks are completely green and completely mucky. And you look down in those are the control tanks. And then you look in the predator removal tanks and they're crystal clear. Absolutely no primary productivity whatsoever. So there is a trophic cascade affecting that. I just didn't measure it.
Cool, thanks.
Regrets from my dissertation.
So we have plenty of systems where top predators have been removed not because of catastrophic death, but because of specific human activities. And both the terrestrial and marine systems. Has anybody, or have you thought about applying these sorts of hypotheses testing to those systems where it's more manipulative experiment that along the lines of the field data that you got for the dry eye?
Yeah, for sure. I think that us thinking about top predator extinctions as a biological disturbance, that counts. That's a disturbance too. And it can have very similar effects. And in a lot of those systems, It may not be brine that's the concurrent abiotic mess that's going on, but there are multiple threats. And so I like to think that we can do a similar thing with all these other systems. Unfortunately, in a marine system, like the one where you work, we know so little about those abiotic factors, and so we're limited with our field studies. I think in these fall tangible systems, the field work can get us closer, those observational studies.
I guess what I was thinking was that in some of those systems, the top predator removal is dissociated from any abiotic influence, so you only have top predator.
Yeah, that's true. And that was, actually, I started with the top predator removal study because in the background of an extreme abiotic environment that may not be changing, that top predator effect is still strong. Yeah.
Okay, the stained glass, that's beautiful. And that raises the issue of a trait type that you didn't focus on, but I wonder what you think about reproductive trait types. It seems like there might be some substantial differences in reproductive behavior.
I wish I'd thrown in a really awesome photo of the eggs. So the reason that they have lost flight, the reason we believe that they lost the capacity to fly, is because all of those flight muscles have been reabsorbed and turned into reproductive tissue. And that trade-off has allowed them to create enormous eggs. So the entire back of this water bug is covered like this in eggs. And they're huge. They're the size of Tic Tacs on a bug that's this big. Right? So that's a huge investment of time and resources. And I don't remember your question. I got so excited about it.
So given all the species that you have in your streams, arid streams, do you think the reproductive behavior, reproductive traits, like you described, the variants could be interesting in the same type of analysis?
Oh, definitely. And I tried to get it out with the limited history characteristics that we have. We just don't measure that usually for these tiny invertebrates. But it's definitely associated with their dispersal capacity and their capacity to recolonize following disturbance. I mean, the giant water bug is not only flightless, but it needs huge amounts of resources in order to reproduce, even if it were to get to one of those novel post-disturbance habitats. Good question. Yeah, Becca.
Following up on the traits thing, so the ordination analyses you did also allow you to sort of flip it and look at what traits are loading most heavily on the different axes. And so I'm wondering, were there any common traits that showed up as imparting sort of resilience to this type of disturbance, or other traits that showed up as being like a red flag that this species is going to have a tough time?
Body size was associated with a tough time. I remember that was really high association with one of the axes.
Big or small?
What?
Big guys have a hard time? Or
Big
small
guys have a hard time. Yeah, I mean, that's supported by a whole bunch of literature now. So the top predator and that detritivore are both threatened and both large bodied. But in addition to that, there weren't a lot of consistencies. Dispersal capacity for the movement traits, definitely for the recolonizers, for the resilience. But besides that, that's all the pattern I can remember.
Okay, well, I think it's time for the events of North Elmwood. Thanks again, Kate.